Okay, how exciting! Now we get to talk about secondary structure. So as you remember from Biology 115 and from your other chemistry classes, that secondary structure, we're talking about the alpha helices and the beta strands. And these are the abbreviations. So the alpha helix looks like a helix, the beta strand looks like an arrow. There's also the beta turn, which we're not going to go too much in, and then also the sort of random loop. So those are all the options for secondary structure. So after watching this lecture, you should be able to name the main contributions of forces that stabilize secondary structure. You should be able to compare the relative stability of different helices, beta strands, beta sheets, and interpret a Ramachandran plot. Okay, so let's dive right in. Oh, vocabulary note. Beta strand means one. Beta sheet means more than one. So you have multiple strands interacting, typically more than two, really. Okay. So first off, let's go back and look at the amino acid structures themselves. So here we have the peptide bonds shown in yellow, and what you notice is that this carbon has a carbonyl. What that means for amino acids is that this bond is essentially planar. Similarly, the next peptide bond is also planar. These aren't on the same plane, they are at some angle to each other. And that angle is because the alpha carbon is in between these two, and this plane can rotate this direction or the other direction, and we can rotate around this bond as well. But essentially this is stuck because of the planar nature of carbonyls. This double bond makes it stuck. So we define the angle that this plane rests in with the N compared to the C as the phi angle. The next angle we refer to as the Sai angle. The Sai angle is by the carbonyl. The Sai angle is by the C. The phi angle is by the N. So it's like a fin and then the C. Fin and the C. Okay. And so depending on the identity of the R group, is it big and bulky? Is it small? That's going to change the angles here. And so each amino acid has its own phi and psi angles that it's allowed to be in before steric hindrance occurs. Okay. And so based on those phi and psi angles and the patterns, so psi is here, so this is positive psi, negative psi, we can make a Ramachandran plot. And what this is named for a person, Dr. Ramachandran, who discovered that you can predict whether something's going to be in a helix, a beta sheet, left-handed or right-handed, based on these phi and psi angles. This was a huge discovery, and it shows the predictability and the real chemical basis of all of the things that we see in biochemistry. There's a chemical basis based on steric hindrance that's predictable, that's relatable. Okay. So we see beta sheets occur where you have a positive psi angle here and a negative phi angle here. Right-handed helices occur at a negative psi and a negative phi. And then left-handed helices occur at positive psi, positive phi. So what's the difference between a right-handed and left-handed helix? And this is a very conceptual question that you might have to struggle with for a little bit, but we're going to work on models during class. So a right-handed helix, imagine a spiral staircase. And as you are walking up the spiral staircase, your right hand is on the rail all the way up. That would be a right-handed helix. However, if your left hand is on the rail all the way up, that would be a left-handed helix. Okay, so we're going to work more on that during class as well. And then remember, beta sheets are more than one beta strands. all together. Okay. So what about the individual interactions? What forces keep these together? And for both of them, the answer is hydrogen bonds. In helices, we see these hydrogen bond interactions about every 4 or 3.6 or 4 residues. And so we see hydrogen bonding between the carbonyl carbon and the polar hydrogen attached to the amino group. And so what that looks like, I'm not very good at drawing this, 2, 3, and this is kind of 4, kind of looks like this. So we've got hydrogen bonds stabilizing this, about every 3.6, about every 4 amino acids. In the beta strands, we really see this in the sheets. So we would have some interactions between the carbonyl carbon and the polar H attached to the amino group. And so we'd have hydrogen bond interactions that are actually quite distant in the primary structure, because there could be any sort of thing in between these 2 points. Correct? Correct. I have a PhD. So for beta strands, this kind of gives us another layer of complexity, where we can have beta strands that form sheets where the strands are oriented in the same direction from N terminus to C terminus, and we call that parallel, because all of the N's and all of the C's line up. When they do not line up, N to C, N to C, we call that anti-parallel. Okay. So you can imagine, in order to get a parallel structure here, we'd have to have something occurring and in between, and then it goes back down this way. Whereas for this, we could just have a loop going straight that way. Okay. So you have to have some other structure in between to get a parallel strand. Whereas anti-parallel, it can just keep going. Basically, it could just fold over with a turn or what have you. But now we need to compare the stability of these two. And I'm going to erase this, don't mind. Okay. And so what we see for parallel sheets down here, the hydrogen bonds are shown in green in this figure. For the anti-parallel sheets, the hydrogen bonds are shown in red. So we need to figure out what's more stable, the parallel or the anti-parallel. So how would you figure that out? You would count the number of bonds over the same number of residues. So as you can see, in the parallel, these are offset. So you actually end up with fewer parallel, only 1, 2, 3, 4, 5, compared with 1, 2, 3, 4, 5, 6 hydrogen bonds in the anti-parallel. So even though hydrogen bonds are very weak, there's more in anti-parallel, therefore it is stronger. Okay. So now we're going to talk about amphipathic helices and sheets. So amphi means both. Pathic refers to the charge, so we see both charges, sort of, right? So these are, they'll have polar and nonpolar residues in them. That's what amphipathic really means. And so if you're looking down the barrel of a helix that's amphipathic, what you would see is that one side would have one charge and the other side would have either no charge or a different charge. Okay, that's what makes it amphipathic. Okay. So a lot of times this would be like something polar and something nonpolar. Okay. So when we have a bunch of amphipathic helices, some proteins use that as a way to get through the membrane. These are all helices looking from the top, and we can have a charge on the outside and a negative charge, or have nonpolar regions on the outside rather, and polar regions on the inside, and this basically makes a tunnel that something with a charge can get through. Okay, so how does this happen? So for amphipathic helices, remember that we're repeating every 4 amino acids about. So every 4th amino acid would be polar, and that would create one side of this having some charge and the other side not having any charge. For sheets, this is different because if you draw it out, you kind of get alternating side chains. One side, the other side. Oh, ignore this. R. They kind of face every other direction. So this would be every other amino acid is polar, and that would result in an amphipathic beta sheet. So you end up with a charge on one side and a nonpolar on the other side. Okay, so I think that's it. Yep, that's it.